Okay, hello everyone. Welcome to the Fertility Basic Science Committee webinar. I'm Dr. Xiao, an assistant professor from Rutgers University School of Pharmacy. I'm a co-chairing the Fertility Basic Science Committee with Dr. Ariella Shikova from University of Michigan. And today we are very excited to have Dr. and also Professor Karla Hutt for giving a webinar. So I will just give a very brief introduction about Dr. Hutt. Dr. Hutt is an NHMRC investigator and deputy head of the Department of Anatomy and Developmental Biology, and also a co-head of the Development and Stem Cell Discovery Program at Monash University. And she obtained her PhD from Australian National University in 2006, where her research focused on understanding ovarian development. She then did her postdoc research at the University of Kansas Medical Center, or KUMC, in the United States. And her postdoc research focused on the impact of environmental toxicant on oocyte biology and also embryo quality. In 2008, she returned to Australia to work with Professor Jock Findlay as the NHMRC Peter Doherty Fellow. And in 2015, Dr. Hutt started her own lab at the Department of Anatomy and Development Biology at Monash University, and she is now leading the ovarian biology laboratory. And currently, Dr. Hutt's lab research is primarily focused on studying the biological, cellular, and molecular mechanisms underlying the loss of oocyte number quality associated with reproductive aging, environmental contamination, cancer treatment, and also infection. So her talk title today is Defining and Mitigating the Impact of New Cancer Treatment on Ovarian Function and Fertility. Without further ado, I will turn it over to Dr. Hutt.
Great, thank you so much for that lovely introduction. So I'm very pleased to be here today to speak to you about the work that my lab has been doing towards really trying to understand the impact of some of the newer cancer treatments on ovarian function and fertility. So I will start with a very brief background. I understand that this is not progressing. I might stop the share and reshare because it's not progressing.
Sure, you can just reshare, no problem.
Here we go. We all good now? Yes. Okay, so just to start with a little bit of background in case there are some people that are new to the field or some new trainees or students in the audience. We know that cancer diagnoses are on the increase worldwide. And just as an Australian example, 1 in 7 women are diagnosed with breast cancer during their lifetime. So that's a really, really big number. But fortunately, improved treatments and early detection now mean that cancer survivorship is higher than ever before. In fact, the 5-year survival rate now exceeds 80% across many cancer types. So, but we do know that cytotoxic chemotherapy and radiation can cause collateral damage to the ovary. And if that damage is severe enough, then that can lead to infertility and premature menopause. And premature menopause has a lot of negative health consequences associated with it. And I've just listed some of those here on this slide, including an increased risk of mental health disorders, increased risk of glaucoma, dementia, cardiovascular disease, osteoporosis, decreased sexual health, and an increased risk of early death. And that really means that preserving fertility and preventing premature menopause are really critical for ensuring the long-term health and also the mental well-being of cancer survivors. So the risk of ovarian damage is really highly variable, and it really depends on the type of treatment that is used. So with the chemotherapies, these are typically divided into chemotherapies that are high risk, moderate risk, or low risk. And it's really the alkylators that have proven to be the most damaging to the ovary. And that's probably because they are able to target cells at all stages of the cell cycle and induce DNA damage, which means that they can directly damage the DNA of oocytes. With radiation therapy, it's all about location, so radiation delivered close to the ovary, such as to the pelvis, abdomen, or spine, or total body irradiation can be very harmful. And of course, there are these contributing factors like age, the dose of the treatment, and what combination treatments are being used that need to be considered. But fortunately, as a result of a lot of basic discovery research as well as clinical research over a number of years, we do now have some really good fertility preservation options available, including the cryopreservation of embryos, oocytes, and ovarian tissue. And I'm not going to go into the details here, but whilst these are very good options, each of these options does have limitations and are not suitable for all women. And that's really driven new research to increase fertility preservation options. And that's part of what many of us in this group do. Now, my lab is particularly interested in the development of pharmacological ovary protectants. And by that, I simply mean drugs that can protect the ovary from damage caused by cancer treatments to preserve fertility and also endocrine function. But if we are going to do this, we really need a much, much better understanding of which cancer treatments are risky and precisely how different cancer treatments compromise fertility. So this slide here is really just a schematic describing the approach that my lab takes to fertility preservation. So firstly, we're defining the impact of new and existing cancer treatments on ovarian function and fertility. We identify the cellular and molecular pathways that trigger follicle depletion. And then we set about to determine whether blocking these pathways can actually prevent follicle loss and preserve fertility. We also spend a lot of time determining whether or not this is a safe thing to do, whether keeping oocytes and follicles that have been damaged from dying, whether keeping them and using them is really safe to do. So we've done a lot of studies on oocyte quality and offspring health. We've also moved towards testing and optimizing some small molecule ovary protectants. And those of you that know me well will know that I'm very focused on the ovary, but actually more recently we've turned our attention to also considering the uterus in this picture. And so we've been characterizing the impact of cancer treatments on the uterus with the view to identifying novel uterine protectants. How do we do this? We typically harness the power of preclinical models. I'm not sure about others of you in this call today, but often I get asked by our clinical collaborators why they should care about studies in mice. And so this is a really important question to be able to answer. So firstly, we can undertake properly controlled studies that are specifically designed to determine the impact of cancer treatments on the ovary and also the uterus. We can test single agents at a time. We can very accurately and rapidly assess follicle depletion and ovarian damage. And we can tease apart mechanisms and we can test protectants. And it's simply not possible to do these things in women. And we can actually generate data years before fertility outcomes in women receiving some of these new drugs become available. And until that clinical data becomes available, we can use this data that we generate in mice to help advise clinicians and patients to provide a better understanding of the potential risk and benefits of different treatments. And this can really help to assist with informed decision making. For example, are these the right treatments for me? Should I consider fertility preservation? So I started my lab in 2015, and right from that point, we started looking at a variety of the different traditional treatments. So these tend to be the genotoxic and DNA damaging agents. And we've looked at a range of these treatments, including radiation, cyclophosphamide, and cisplatin, but also some others about which less is known, like 5-FU and vincristine. So for many of these, we've determined the extent of ovarian damage, and for some uterine damage. We've started to identify mechanisms, and we're now testing novel protective drugs. I'm not actually going to talk any further about that particular work today because that's a whole seminar on its own. But what I am going to talk about is some of our work looking at the impact of new treatments on the ovary and fertility. So this work has been done in collaboration with Shireen Loy, Kelly Phillips, and Wanda Chu. And they are medical oncologists at the Peter Mac, which is a cancer research, education, and treatment centre here in Melbourne. And this work has really been driven by patient requests for information. And there's two particular classes of drugs that we've been looking at. The first, PARP inhibitors, like olaparib. So olaparib is a new drug that's being used to treat women with BRCA1 or 2 mutations who have ovarian or breast cancer, and also the immunotherapies. So the landscape of cancer treatment is really transitioning towards these targeted treatments like immunotherapies. And that's because these treatments promise improved outcomes with reduced side effects, which sounds wonderful. There are lots of different types of immunotherapies, and the one that I'm going to talk about today are the immune checkpoint inhibitors. So immune checkpoint inhibitors are really important. They are now standard of care for numerous cancers, including in the curative setting. And they're probably most well known for their very effective treatment of melanoma. So people that used to die from melanoma are now surviving as a result of immune checkpoint inhibitors. But they're also now being used for a range of other cancers, including triple-negative breast cancer, cervical cancer, and lung cancer. And these are all cancers that do occur in reproductive age women. So what are immune checkpoints? So there are 3 molecules that I'm going to talk about called PD-1, PD-L1, and CTLA-4. So these are all immune checkpoint molecules. And immune checkpoints are super important because what they do is they turn your immune system off. So they prevent your immune system from attacking other cells or tissues in your body. So they're very important for generating this self-tolerance. So if we just look at this diagram down the bottom here, we can see that there's a T cell in blue, and that's expressing PD-L1, and there's a tumor cell in gray, and that's expressing PD-1. And that interaction actually tells that T cell that this is a normal, healthy, happy human self cell, and to leave it alone. But what immune checkpoint inhibitors do is they're monoclonal antibodies that prevent this interaction from occurring. So they enable the immune system to attack cancer cells, and they do this by enhancing the activation and proliferation of these cytotoxic T cells, so they're the T cells that can kill other cells, but they also upregulate circulating inflammatory cytokines. So one of those is TNF-alpha, which I'm sure many of you are familiar with. Interestingly, however, these immune checkpoint inhibitors were introduced into the clinic, but the impacts on ovarian function and fertility were never investigated. So why is it important that we do this work? Well, we know that reproductive endpoints are really included in clinical trials for any cancer treatment. And if you want some evidence for this, a collaborator of ours conducted a systematic evaluation in the context of breast cancer, demonstrating that really ovarian function endpoints are hardly ever included in any clinical trials. There were, in fact, no studies assessing ovarian toxicity of immune checkpoint inhibitors before they were used. And that really means that real-life data on the fertility of women being treated with these new drugs won't actually be available for years. Young survivors do report infertility to be almost as devastating as their cancer diagnosis. Some women will modify their treatment in order to avoid infertility, and women have very clearly told us that they want to know the risks before they start their treatment. They want to know if they'll be able to have children. They want to know if they should utilize fertility preservation before they start on the immunotherapies. They also want to know if perhaps there's a treatment that works just as well but won't compromise their fertility. So this is just a schematic diagram of an ovary, which you'll all be familiar with. And I've put this here because this was part of our thinking about why we thought immunotherapies might actually have an impact on the ovary. So we've got our primordial follicles or ovarian reserve around the cortex of the ovary. We've got our developing follicles leading to ovulation, and then the corpus luteum. But the ovary also contains lots of blood vessels, lots of immune cells like T cells shown here in light blue, and also cytokines shown here in dark blue, and it's just an example of TNF alpha. And those immune cells and cytokines are actually really important for normal ovarian function. And in addition to this, we know that alterations in immune cells and cytokines can impact on oocyte number and quality. So this really led us to hypothesize a couple of years ago now that immune checkpoint inhibition might impair oocyte number and quality by altering the local immune environment. And the aim of these studies was really to evaluate the impacts of anti-PD-1, anti-PD-L1, and anti-CTLA4 on ovarian function. And to do this, we use two different types of mouse models. We use a breast cancer mouse model, but also a healthy mouse model. And that's primarily because there are some limitations as to what we can study in the breast cancer mouse model. And that's because these mice do have a tumor and they tend to get sick, and ethically we cannot continue with the study. So I'm just going to show you some of the highlights of the data that we've generated so far, primarily from the healthy mouse model, because as I said, we can get a bit more information from those. So this is just an example of the experimental design that we use. So we start with adult C57 black 6 mice. We inject them 3 times on day 1, 4, or 7 with either an IgG isotype control or an immunotherapy like anti-PD-L1, anti-CTLA4. We take ovaries from the mice a day after the final treatment to look at very early stages of what's happening in there. And then we also have a look at 21 days after the final treatment. We monitor their estrus cycling, and we also superovulate the mice to look at oocyte yield and oocyte quality. So just starting with my favorite thing, which is to count primordial follicles and ovaries. So what you're looking at here is a graph of primordial follicle number and primary follicle number in mice that have been treated with either the IgG control, anti-PD-L1, or anti-CTLA4. And this is at the 21-day time point. And you can see that there is a really dramatic decrease in the number of primordial follicles that are present in the ovary of these mice at this 21-day time point. What about the growing follicles? Well, it's at the 21-day time point, we don't actually see any significant difference in the number of healthy secondary or antral follicles, but we do see an increase in the number of atretic follicles that are present in the ovary, suggesting that checkpoint inhibition can impair the survival or induce the atresia of these growing follicles. Interestingly, when we look at the early time point, we actually do see a significant depletion in the healthy growing follicles as well. So it seems as if the first evidence of effects on the ovary are in fact a depletion of the healthy secondary and antral follicles, and then later on we can see a depletion of the primordial follicles. So what about corporal lutein number? We often use this as a surrogate for ovulation. And even just looking at these cross sections of the ovaries, you can see that the anti-PD-L1 and anti-CTLA4 ovaries do look quite different to the control mice. They look far less active, and it looks as if there are less corporal lutein there. And when we quantify that, we do indeed see that the corporal lutein number is depleted after checkpoint inhibition, suggesting to us that this types of treatment could perhaps impair ovulation. As I mentioned, we do superovulate the animals and then look at the number and the quality of those oocytes that are ovulated. With the anti-PD-L1 treatment, we tend to observe that there are normal numbers of oocytes ovulated, but actually a lot of them are fragmented and dying. And with the CTLA4 treatment, we do see that there are fewer oocytes that are actually ovulated, suggesting that checkpoint inhibition can impair both the number and quality of ovulated oocytes. So the last reproductive endpoint that we've looked at in this particular study was estrus cycling. And so I'm showing you here representative plots from our 3 treatment groups, our IgG, our anti-PD-L1, and our anti-CTLA4. And you can see in the IgG treated control animals that they really undergo estrus cycling very normally. They progress through all the stages as you would expect, and all of the mice did this very normally. However, we do see significant disruption of estrus cycling in the immunotherapy treated animals. And this suggests to us that there could be a possible endocrine disruption that is preventing this estrous cycling from occurring normally. So we're seeing, I guess, a lot of different things happening within the ovary. So we're really keen on trying to define the mechanisms by which this is occurring. And a few slides ago, I told you that immune checkpoint inhibitors work for treating cancer because they cause an activation and I guess an upregulation of immune cells and also cytokines. So one of the things that we did early on in this study was to look at immune cell populations in the ovaries. And so what I'm showing you here is actually just an example from our tumor-bearing mice at an early time point. And you can see that the number of CD4 positive T cells, as well as CD8 positive T cells, and the subset of those CD8 positive T cells that are PD-1 expressing are all upregulated. There's more of them in the ovaries after treatment with immunotherapies. So checkpoint inhibition actually expands the number of ovarian T cells that are present. So are these cells necessary for the impacts that we're seeing on the ovary? So one of the ways that we sort of have started to investigate that question is to use RAG2 knockout mice. So these are mice that don't have mature T cells, they don't have mature B cells, they also don't have natural killer cells. And so when you treat these mice in exactly the same way as we've done for all of our other studies, what we find is normal numbers of primordial follicles, normal rates of follicle atresia, and also normal CL numbers. And so this suggests to us that the immune checkpoint inhibitor-induced ovarian dysfunction and follicle loss actually requires immune cells. We don't know from this study which subset of immune cells is critical, so we are still working on that. But what about cytokines? We were very interested to know whether there was local cytokine production within the ovary. So to begin to answer that question, we just did a very simple quantitative PCR on ovaries from our control and treated animals, looking at a range of cytokines that had been shown in the cancer field to be upregulated in response to immune checkpoint inhibitors. And actually we found that there was a trend towards increase with all of the inflammatory cytokines, but this was significant with TNF-alpha. And actually, when we look at TNF-alpha levels in the serum, we find that this is indeed elevated at 21 days. So elevated intra-ovarian and circulating TNF-alpha levels may actually contribute, at least in part, to the checkpoint inhibitor-mediated follicle loss. So we actually became pretty interested in TNF-alpha. This is a pro-inflammatory cytokine that triggers apoptosis through signaling death receptors and actually activates the extrinsic apoptosis pathway. So we wanted to know whether TNF-alpha just on its own can induce depletion of primordial follicles and impair oocyte quality. So we just did a couple of very simple experiments to get some insight into this. So firstly, we just cultured some neonatal ovaries. I think they were postnatal day 5 or thereabouts, either in the presence or absence of TNF-alpha. And what we found was that the addition of TNF-alpha to the ovary culture media actually induced the death of oocytes in primordial follicles. And we were able to confirm that this was a very specific effect mediated by the extrinsic apoptosis pathway by repeating the experiment using BID knockout mice. That is a protein that is important for death signaling by TNF-alpha, and we were able to mitigate this impact. We also added TNF-alpha to IVM culture media, and we found that there was a slight but significant decrease in the ability of oocytes to mature to M2. So this kind of indicates to us that even a small change in just one cytokine can potentially impact how the ovary functions. I'm just going to very briefly summarize all of those key points and key outcomes from this study. Firstly, there's a depletion of primordial follicles, a degeneration of the growing follicles, a decreased number of corporalia, reduced number and quality of ovulated oocytes, disrupted cycling, potentially impaired endocrine function. And in addition to this, we do see increased intra-ovarian T cells, CD4 positive and CD8 positive T cells, as well as TNF-alpha, which could be the mechanism underlying all of these issues that we are seeing within the ovary. So immune checkpoint inhibition could actually have quite far-reaching implications for the health and fertility of women. And in particular, the depletion of the ovarian reserve can have permanent consequences for fertility and endocrine health, as I mentioned at the very beginning of this talk. And in fact, the impact of immunotherapies on the ovary might not actually be better than some of those traditional cytotoxic chemotherapies. And certainly we should be starting to think about fertility preservation for women undergoing these treatments, at least until we can obtain some clinical data. So this is still ongoing work. What we're doing now is we are conducting breeding studies to assess fertile lifespan and impacts on pregnancy, establishment, and maintenance. And we do have some early data to suggest that there are some impacts on pregnancy as well. We are really keen on working towards defining the mechanisms of checkpoint inhibitor-mediated ovarian damage. We do think it's immune cell and cytokine mediated, but we are studying this in more detail with some of our immunology collaborators. We are testing protective strategies, and in particular, we're looking at TNF-alpha inhibitors, one of which is infliximab, which is clinically used currently to reduce kind of inflammation, and whether or not that can mitigate some of the impacts of immune checkpoint inhibition on the ovary. But all of the studies that we've done so far have been on mice, but we are interested in, as far as possible, determining how some of these treatments might be impacting ovarian function in women. So we're currently in the process of evaluating serum AMH levels and cytokine levels in a cohort of women. We have about 200 women in the study that have been treated with immune checkpoint inhibitors. So I've got two thought bubbles to share with you today. This is the first of my thought bubbles. Firstly, oocytes are very sensitive to their environment, and we know this, but it seems that the immune environment is really emerging as a major player in the regulation of oocyte number and quality. And even really subtle changes in the immune environment can potentially have major impacts. And so this has implication not only for women that are receiving immunotherapies for cancer treatment, but also for older women in which the ovarian microenvironment is transitioning towards a low-level chronic kind of environment. Also women with infections and those with inflammatory diseases. And we actually have projects in the lab focusing on all of those things now. Okay, so that's the end of the immunotherapy story for now, and I just want to share with you a little bit of the data that we've generated on the impact of PARP inhibitors on the ovary. And this work was really stimulated by Kelly Phillips, our clinical collaborator. She called me a number of years ago. She had a number of women that were being treated with olaparib, a new cancer drug at that time. And those women were asking whether this drug might impair their fertility. And as an oncologist, a medical oncologist, Kelly didn't have any information to provide them. So she called me and she said, can you help us figure out whether or not there might be any potential risks here? And at that time, I kind of had a look at PARP inhibitors and how they might work, and I thought it was probably a pretty low risk. So we didn't actually proceed with any mouse studies at that stage. But then a year later, she called me again and said, look, my patients really want some information on this. Can we just do a small study to see whether PARP inhibitors might be harmful to ovaries? So PARP inhibitors, or PARP proteins, are proteins that help repair DNA damage, in particular single-strand DNA breaks. And PARP inhibitors impair the ability of cancer cells to repair their DNA. And this is thought to sensitize cancer cells to apoptosis after treatment with genotoxic drugs. So that's basically how these new drugs are thought to work. So for our mouse study, we took wild type C57BL6J mice that were 8 weeks of age, so adult mice, and we gave them 28 daily subcutaneous injections with either vehicle or olaparib. And actually women receiving PARP inhibitors do receive them for many months at a time. So this is a relatively short treatment. So we looked at all of our kind of usual things that we do when we analyze ovaries. We found that there was normal estrous cycling. normal ovulation, normal number of growing follicles, normal serum AMH, so everything was looking really good. But quite surprisingly, we did find that olaparib depletes primordial follicles by around 46%, so we were not expecting that. So what are the implications of this small mouse study? Well, PARP inhibitors are set to become more widely administered, so more women will be receiving them. And women treated with PARP inhibitors based on our mouse studies could potentially present with normal AMH levels and cycling normally, but unknowingly have reduced ovarian reserve. So this treatment is one that is used for women with BRCA mutations. So that means that they already have a germline DNA repair defect and potentially a reduced ovarian reserve. And so the impacts of PARP inhibition on the ovary could actually be more serious in women with a BRCA1 and 2 mutation. So certainly the study suggests to us that much more work is required. And our collaborators are now looking at measuring serum AMH in women before and after olaparib treatment. And we are now working towards testing the impact of olaparib treatment in BRCA mutant mice to see if there is, in fact, an exacerbation of effects. Okay, so just for the final few slides of my talk today, I wanted us to think a little bit more broadly about the impact of cancer treatments on fertility and move away a little bit from the ovary and start to think about the reproductive tract more generally, and in particular the uterus. So we know a lot about the impact of different cancer treatments on the ovary, but we know much less about cancer treatments and the uterus. But if you do a really, really deep dive into the clinical literature, there are some hints that radiation in particular, but also some chemotherapies might actually have a really negative impact on the uterus. In particular, there is some evidence of reduced endometrial thickness, smaller uterine volume, lower pregnancy rates, increased spontaneous abortion rates, even when donated oocytes are used, and more embryo transfers tend to be required to achieve pregnancy. In addition to this, there is some evidence that birth outcomes are impacted, like preterm birth, low birth weight, and small for gestation age. But actually, from these clinical studies in women, it's really not possible to tease apart what we're seeing as a direct uterine damage effect versus impaired uterine function due to reduced endocrine support resulting from this ovarian damage that we know occurs, or from a reduction in oocyte quality. So to get around this problem, we started to develop a mouse model that allows us to determine what is the result of damage to the uterus, independent of any impacts on the ovary. And I'm not going to explain this model in detail because it's quite complicated and it's all published, and I've got some references that you can look up if you need to see it. But essentially it involves transferring healthy embryos from donor mice into recipient mice that have been exposed to radiation. So firstly, what we've been able to show so far is that we can detect a lot of DNA damage and a lot of apoptosis in the uterus after irradiation. So we just have looked at gamma H2AX staining in various compartments of the uterus, as well as tunnel to look at apoptosis. And we do see a lot of it there after irradiation. And when we transfer those healthy donor embryos into those recipient mice that have been irradiated, we actually see that implantation occurs normally. We get a normal number of implantation sites, and the sizes of those implantations look normal. But when we looked at the mouse uteri, they do look a bit different. They tend to be a bit whiter and a little bit more atrophied after radiation, which indicated that there was a potential problem. And in fact, when we looked 10 days post-embryo transfer in the irradiated mice, we saw significant resorption of the implantation sites, which is quite dramatic to see, and you can see it here in these two images. And when we've tried to provide a little bit more insight into the mechanism, it does seem like there is uterine artery endothelial dysfunction, as well as impaired decidualization. And so this is work that has been led by Dr. Amy Winship, who is a group leader in the lab, and our PhD student, Megan Griffiths, who has subsequently graduated and is now doing her postdoc. So all of this work to date has been published. So if you're after some more details, there's some references here at the bottom of this slide. So I guess this has really inspired us to think about fertility preservation a little bit differently and take a new approach, because we think that possibly to maximize the chance of pregnancy success, fertility preservation agents now that we're looking at should really try and protect both the ovaries and the uterus. So this is my second thought bubble for the day, and that's the more we learn about the mechanisms underlying cancer treatment-induced infertility, the more likely we are to be able to develop really effective strategies for preservation of ovarian reserve, endocrine function, and also the uterus. But we think that understanding the biology is really essential to this process. So this is my last slide for the day before the acknowledgement slides. And I just wanted to end with this because I know that this is a sort of a basic science group, but I wanted to encourage everybody in their work because their studies in mice can really have positive impacts on women. And I've just got a few examples on this slide here. So firstly, preclinical mouse data can be used to inform women about the potential ovarian and uterine risks associated with specific cancer treatments when there isn't clinical data available. And just as an example of this, our preclinical data demonstrating that olaparib damages mouse ovaries is now actually provided by AstraZeneca to women with breast cancer who are enrolled in clinical trials involving this new drug. In addition to this, the preclinical mouse data on the unexpected impacts of new cancer drugs on ovarian function that we have generated and others potentially in this audience have generated has actually been used to help lobby for the inclusion of ovarian function endpoints in clinical trials. So this has actually resulted in the first international recommendations for measuring ovarian toxicity in all cancer drug trials that enroll post-pubertal premenopausal women. And this was a research statement published by ASCO at the end of last year. So that's quite an exciting area of progress in the field. Okay, so a lot of people have been involved in the work that I've talked about today, particularly Lauren Alessi and Amy Winship, who produced all the immunotherapies work, as well as the olaparib work. I work with a big team of students and trainees that contribute to everything we do. At Monash University, we're also lucky enough to have excellent technology research platforms. So these are platforms that have expertise in almost any technique or technology that you can think of, and you can go and get support and help from them. So we use them for a lot of our work. We also have a lot of collaborators that we work with, including the medical oncologists that I've mentioned a few times, immunologists, apoptosis experts, and also those that are doing the more translational human work that we do, in particular Richard Anderson and Roseanne Rosario, who collaborate with us on some of our novel protectant studies. And that is it for today.
Thank you, Karla, for such a wonderful talk and also great research. We already have several questions in the chat box. So for all participants, you feel free to type a question in the chat box or unmute yourself. I'll first read 3 questions in the chat box. So the first question is, what is the exact age of adult mice used in the immune checkpoint inhibitor project, the first project you mentioned?
Yeah. So they receive their first injection when they're between 7 and 8 weeks of age. And then they will be treated for approximately 2 weeks after that. We'll do our first analysis 24 hours after that, our second one 21 days after that. We've actually also done a 100-day time point as well. And of course, we still see that the primordial follicular ovarian reserve is depleted, but those impacts on the growing follicles are no longer evident, and ovulation appears to be normal at that time as well. In addition to that, all of those kind of immune indicators that we're looking at, including the T cell levels in the ovary and the cytokine levels, have returned to baseline by that time.
Got you. Thank you. We can take turn between the chatbot question and also the lab question. So do we have any question online? Okay. The next question is, how does the 50 milligram per day per kg in mice compare with the olaparib treatment women are receiving?
Yeah, so I think that's an excellent question, and it's something we always struggle with our mouse models is trying to, I guess, replicate in some way what happens in the clinic. So I actually can't remember how we selected that study, but what we usually do is we try and look at the clinical regimen that women are getting and then kind of scale it down milligrams per kilogram to what a mouse is. And then the time frame and regimen, we usually try and replicate at least one cycle. Sometimes we do more cycles, but for the olaparib, it was just the 28 days. So women can receive olaparib for up to a year. Obviously, we're not going to treat mice for a year, but we felt that we should see results if anything is happening within 28 days. But potentially, that's actually even a truncated regimen in a mouse, and if we did that for longer, we might even see more severe effects.
Got you. So Leslie, I see you have a video on. Do you have a question? Leslie. Okay, got you. You don't have to turn off your. Yeah, sure.
With the speakers when they can't see anybody when they're answering questions. So
Sure. Yeah, thank you. Yeah. So we have a nice question. Yeah, Jasper said thank you. I know she is a PhD student from Dr. Lin Keating's lab. And we have another question from Dr. Qiang Zhang from Emory University. Do T cells normally participate in the atresia of growing follicles and also primordial follicles?
Also a fabulous question. So when we started this work, we are not immunologists at all, but we've been kind of taking a crash course in immunology and started to look into the literature around kind of what immune cell populations are present in the ovary, what's important, what's not. And actually we were pretty surprised when we started that there was very little known about specific immune cell populations in the ovary and what their roles might be. There's a little bit about macrophages, not a lot about T cells. Actually, since we started this study, there has been a couple of publications that have come out that have characterized immune cell populations across the estrous cycle in mice. I don't think we specifically know what the role of T cells is in follicle atresia, but I think that they're very likely to play a role. I guess it's
thanks. Leslie, go ahead.
Yeah.
So Karla, I do actually have a question. Thank you for your presentation. In looking at the uterus and the injury that's conferred from radiation, what therapies have you started to explore? I don't know if that's too early in your research to divulge, but have you noticed? Yeah.
So we've been interested in a number of years at a protein called PUMA. So PUMA is a protein that is part of the apoptotic cascade, and it actually triggers apoptosis in response to DNA damage. And we've done a lot of work to show that it's the key protein that triggers apoptosis in primordial follicle ovocytes following DNA damage. And so we kind of naturally looked at PUMA in the uterus as well because there's DNA damage being induced, and it's likely to be induced by the same pathways. So we've actually replicated some of those studies that I showed you with radiation in the embryo transfer study into PUMA knockout mice that have been irradiated, and that actually mitigates all of those effects. So for us, it's nice because we've been thinking that ideally we do want something that can protect both the uterus and the ovaries at the same time. And if a PUMA inhibitor can do that, then that's where we're going. So we've done all the studies that we've published so far have been on the PUMA knockout mice, so a genetic model. We do have a whole bunch of studies that we are wrapping up right now using a PUMA inhibitor that are really exciting that we hope to publish towards the end of this year. Yeah.
That's fantastic. And do you envision that that's something that would be administered post-treatment, post-chemotherapy, post-radiation, not during, so that you don't affect the actual treatment of the cancers?
Yeah, so that's an excellent question. So far, there's not really any evidence that impairing PUMA will impact on the efficacy of cancer treatment. We've actually done a lot of studies with cancer cell lines, and we're hoping to use a tumor mouse model to also verify that in our mouse models that we can effectively treat tumor at the same time as protecting the ovary and the uterus. So at the moment, we don't see that as an issue, but
That's
we are continuing to work on that.
Thank you. And I have another question. So obviously, anytime we hear that chemotherapy may affect the uterus, everyone gets very nervous, right? And I know there are only about 2 studies that show that. And I believe a lot of that is focused on doxorubicin. You can correct me. What are your thoughts? Like, how worried?
So I don't think there's great cause for concern right now, because I think you probably would have seen it clinically if there was something really significant there. But in saying that the available data is very scant, and they're from patient populations that haven't been designed to specifically answer this question. So there are kind of complicating factors there. So we, in our mouse model so far, have only got very, very solid data for radiation. There definitely appears to be impact there, and I think that's consistent with the clinical literature. We've only just started to look at the chemotherapies, and we have a PhD student that started last year that is very focused on looking at the impact of different types of chemotherapy on the uterus specifically. And at this point, it's not super clear.
Thank you so much.
Yeah, thank you, Leslie. Thank you for bringing up the doxorubicin. We also consistently see doxorubicin can affect the primordial follicle number. Is so interesting. Could be a very important discussion point during the ongoing fertility meeting. So Karla, I have a question about the immune checkpoint inhibitor. So if you see the expansion of the T cells and also elevated cytokines or the inflammation factors, are those residential immune cells or recruited from the system circulation? And no matter what, like locally or from systemic circulation, where are the real target? For example, the cytokine receptors, are they in which state of follicle and also which type of the follicular cells?
Love that question. So that's really kind of what we've kind of focused on for the next few years, is really trying to get some clarity onto, so far we think they're actually resident T cells. I didn't show you some of the data that we've got for that, but it does look like there's a resident population of T cells that are expanding. So then the question is why, which antigen are they recognizing and expanding in response to? And so we do think that there are ovarian antigens that, in the context of checkpoint inhibition, then become available to stimulate the immune cells in the ovary, and then those T cells then target the ovarian antigens. So we're working with our very, very clever clinical immunology collaborators, and they have now become very interested in using the ovary as a platform to identify novel antigens that can expand resident T cells. So that's kind of the extent of my knowledge when it comes to immunology. In terms of the targets of the cytokines, not too sure. There are receptors for TNF-alpha, for example, on kind of follicles at all stages of development. So potentially that can be, actually we do think it's a direct interaction based on our studies with TNF-alpha and the big knockout mice.
Got you. We have our last question in the chat box. Is there any thought on looking at children, adolescents undergoing cancer treatment and fertility, because fertility is a huge concern for pediatric patients and also parents, including pre and pubertal patients in treatment?
Yeah, that is also an excellent question. So, so far in terms of our animal models, we've focused on adults as a starting point, but our intention has been to look a little bit earlier. I think there are likely to be differences between adults and children, particularly with the immune checkpoints, because the immune system matures is immature in children, basically, and matures differently. So I think there are likely to be differences in the pediatric population and the adults. We do have a collaboration with Deb Gook, who's here at the Women's, who has a very extensive ovarian tissue cryobank, so we can do some studies with human ovarian tissue. However, legally, we're not allowed to use any of the pediatric tissue here in Melbourne. It's not something that we are allowed to do, which is a shame because she's actually got quite a large bank of tissue there that could be really useful, but unfortunately it's not something that we're able to do at the moment.
Got you. Yeah, thank you, Karla, again for the fantastic talk and also all the great questions from our participant. And yeah, hope to see you all at the Onco Fertility meeting in this fall. Yeah, thank you so much. Have a great day. Bye.
Thanks, everybody.